So there's one here that said, it's about my A-levels. So do you think studying further maths at A-level was necessary or made you a stronger applicant? No, it's definitely not necessary, especially so for coming to do chemistry at Oxford, you don't need further maths. It is, so some of the maths course does overlap with further maths, so it was quite nice that I'd covered some of the work before, but you'll be taught everything from scratch. So they're never going to assume that you have further maths, but obviously it's nice if it's a bit more familiar to you. And also I just really liked maths at the time, and that's kind of why I chose to do maths and further maths, but it's definitely not a requirement. And then, okay, we have a question. How do you attach the dye to the molecule and what is the dye made of? Are all fluorescent dyes used in similar experiments the same? For example, if testing for a gene of interest when identifying transformed cells in genetic engineering. Okay, so how do I attach the dye to the molecule? I think I then went through and explained that. So what is the dye made of? The dye it's another chemical compound. So it's quite a big, bulky structure. So that's why I didn't draw it out. I just represented it as that little red ball. But it is just, it's just made of lots of carbon atoms, a few oxygen atoms put together to make a chemical compound. But the properties of that compound mean that it's able to fluoresce. So it's all just about the way that that compound is put together means that it has this innate ability that if you shine red light on it, it can fluoresce. So yeah, I just didn't draw that out. And are all fluorescent dyes in similar experiments the same? No, so there's a lot of different dyes to choose from. So I didn't really talk too much about which dye I chose and how I chose it. But you have to choose your dye depending on what you need it for. So there's dyes that come in a whole range of colours, for example. I chose to use a red one, partly because of the microscope setup that we have, and it's a bit easier to work with the red light. But you can range from green dyes are very popular. So yeah, there's a whole range of different dyes, and you would just choose it depending on what you needed. Is A-level maths essential? Again, that's similar to my further maths question. I can't actually remember for undergrad at Oxford chemistry if you need maths. I think it might be a requirement. You might need maths and chemistry to do chemistry at Oxford, but double check that in prospectus and don't quote me. I'm not sure on that one. Okay, we have another question. Are you planning further investigation into what the synthesized drug does to the mitochondria? Yes, so that's a very good question. So obviously the story is not finished. We only have one step, so we know that it's something in the mitochondria. So in the next step is what exactly, because the mitochondria is actually made up of lots of different proteins. So I haven't actually yet answered the question of which protein is it. I've just answered kind of generally it's a mitochondrial protein. So yeah, my next steps is to investigate a bit further exactly which protein is interacting. That's what I want to, that's ideally the target I would like to get to by the end of my PhD. So I've got about 2 years left. And in that, it would be really great if I could actually get to a protein answer, not just a whole structure of mitochondria. So yeah, that's a very good question. Okay, do you feel that the things you learned in your undergrad and master's degree prepared you for working in the labs? Were there a lot of things that you needed to learn on your own? So the undergrad and the master's degree do prepare you really well for going into a lab. The taught undergrad gives you all that kind of theoretical knowledge that you need to call on when you want to make a compound. You need to know what the different reactivities are like of different functional groups. And that's all knowledge you would have got through your undergrad degrees. And then the masters, again, specifically at Oxford, because you have that whole year of research, it really prepares you well for then going into a whole research PhD, because your master's degree is essentially 1 year spent in the lab doing practical work. That prepares you really well. And you go into it obviously not as familiar with what it's like to work in a lab, and everyone around you in the research group you're working in is aware that you haven't had much experience in a lab setting, and so there's a lot of guidance, especially at the start. And by the end of your master's project, you're like you're ready to go. You could go straight into a PhD of research and have all the skills you need pretty much. For my case, it was a little bit different because, like I said, I'm on the chemistry and cells program, so I actually am doing quite a lot of biology. As you've seen from the pictures, I'm doing microscope work, and I'm doing growing cells, which is not something that's part of the chemistry undergrad degree. So that was all new to me. But because of the course that I'm on, I actually got quite a lot of taught lectures at the start of the PhD that helped me out in kind of understanding the biology of things, which was the new section for me. So yeah, in my case, I did have to learn a bit of the biology on my own, but that's because I chose to almost leave the chemistry field and start to move more into the field of biology. But it's definitely doable. So next question, do you think your current research would still be possible if you did not do biology at A level? And how much did they teach you before you started working on the project? So I've covered that a bit, that there was some taught courses to just teach me some general biology. My supervisor has also been really supportive, my biology supervisor, in understanding that I don't have, or when I started, I didn't have the kind of core skills you would get from doing a biology undergrad. And so taught me quite a lot of the practical element of it. But could I do it if I hadn't have done biology A level? Absolutely, yes. I don't think there was anything that I learned at my biology A level that was crucial that I couldn't have later picked up. So definitely, you might find it a little bit harder if you don't have biology A level, but even with science GCSE, you'll know what a cell is, and that's kind of the basic level that you need to then read on and build on that and be able to do a biology PhD essentially. So someone's given me a follow-up question about the dye, going back to that. If the dye is quite a bulky molecule, how can you be sure it doesn't interact with the mitochondrion or the reactivity of the compound itself? So this is a very interesting question, actually, and this was something that did come up during my work. So I did have to be sure that the result I'm seeing is a result of my compound and not because I've got the dye there. So I've done a lot of different experiments that I didn't talk about here, and there's a lot of different ways that you can do to make sure that the result you're seeing is a real result from the compound and not because of having the dye in the way. And so I guess briefly I could just explain one of the things I've done is actually what I've shown here is attaching the dye directly to the compound, but what I actually do is attach the dye in situ. So when I've already treated the compound, the cells with the compound, I add the dye later on. So the compound's already in position and then the dye comes in later. So the dye shouldn't be affecting the position that the compound is in. And by doing that, I can be more sure that it's actually a result of the compound and not the dye. But yeah, that's a very insightful point, and that is something that I had to be careful of with this work. Next question, what does the compound do to the leishmania's mitochondria? So we don't actually know. We know it interacts there, and we know that the cells then die, but we don't know what it does to them. It doesn't. So I've done some studies to try and see if it changes the shape of the mitochondria, and it doesn't. As far as we can see, the mitochondria shape stays the same, but the cells just can't survive in the presence of the compound, and after 24 hours, all the cells are dead. But we don't actually know what's going on. So that's why it's also really interesting for me to do my next step, which is trying to find that protein that is actually specifically interacting with. And if I have that, and I know how that protein works, then I can make I'll have a much clearer idea of what it's actually doing to the mitochondrion. But at the minute, we don't actually know. Okay, some things generally about Oxford. So how many people are there in a college roughly? So it depends massively on which college you're at. At Somerville, there are about 120 undergrads per year across the 3 to 4 years. And then there's, I think, a similar number of postgrad students as well. So there was, at Somerville in my year, there were 6 people doing chemistry. And these are the people that you will have your tutorials with. So I didn't really talk about this before. You're also, some of you might be aware that the Oxford system's a bit different, that you have tutorials alongside your lectures. And you have those in groups of sort of 2 or 3, and those will be with the other people at the same college as you, studying the same subjects. So there were 6 of us, and we were usually just in 2s or 3s for our tutorials. And this goes on to the next question someone has asked, is the tutorial system a large part of chemistry in Oxford? So the tutorial system is just a large part of the way that the teaching is done at Oxford. So you have lectures with your whole cohort, but then you will always have these tutorials which are based around your college and will be just in much smaller groups. So it's kind of the way that the Oxford teaching is done. So yeah, it's a big part. You'll have 1 to 2 lectures, 1 to 2 tutorials a week, sorry, which is just almost close to one-on-one time, I guess, or two-on-one time with your tutor. So it's a really good opportunity to speak to your tutors about to make sure you're clear exactly what you're learning. If you have any questions, anything you didn't understand from the lectures, that's the time to bring it up, and they'll make sure that you're on track and making sure you can kind of answer the questions that you would need to for your exams, for example. So it is a big way of the way the teaching is set up. But yeah, it's a very good system. And then the last question I have, why did you go for Oxbridge? Do you think your experience has been significantly better than those who did chemistry at other unis? So I can't really talk about it being better than other unis because I haven't experienced any other unis, but personally, I really enjoyed being at Oxford. I did really enjoy the fact that the master's year, so the last year, is a full year of research, because that's quite unusual compared to lots of other universities. In your 4th year, so your kind of master's year, you would still probably have some lectures alongside it or some taught courses and some exams, whereas it's very nice, or I really enjoyed the way that it's done here, that in that final year you can just fully focus on research. And for me, I'm sure you can tell, research is something I really enjoy because I'm still in research doing a PhD, and I'd even chosen to do a gap year and do internships doing like lab-based work. So for me, that was, again, something I really liked about the Oxford degree, that full year of research with no kind of lectures and exams to be trying to balance at the same time. Okay, so the question that just came in is, do you think a plant or fungus's biological functions could link to or provide clues that could help us figure out how they can be used in a clinical setting? Okay, that's an interesting question. So could their functions provide clues that could help us figure out how they can be used in a clinical setting? So as far as I know, generally there doesn't seem to be much of, or there's not normally a pattern between what a plant or a fungus is or does and the properties that some of those natural products have. So, for example, you've got the aspirin, which comes from the bark of a tree, and then you've got quinine, which also comes from a bark of a tree, but one of them's a painkiller and one of them is an anti-malarial, so killing a parasite. And those are quite different clinical results coming from, like, both of them from a bark of a tree, which you think was quite a kind of a similar place in nature. So as far as I know, I wouldn't think that actually the specific plant or fungus can tell you much about what it's going to be like in a clinical setting. And that's partly why you need to then have these isolation chemists that are extracting these compounds from plants and testing them against a range of different things. So, for example, the compound that the natural product compound that I use that kills the leishmania has this anti-leishmanial property. When it was extracted, was tested against a whole range of different things, because you actually have no idea when you extract it what, if anything, it's going to be active against. So usually it's tested against lots of different parasites, for anti-cancer properties, anti-inflammatory properties, and there's kind of a standard screen that you would test your natural products against to see what properties it has. So yeah, and in my case, it turned out to have really good anti-leishmanial properties, which is a great result for me. But generally, you have to test against all these different things because you can't really tell beforehand what the properties are going to be.